All roads lead to Rome. As the literal center of the Roman Empire, the city of Rome beckoned people into its walls with the promise of bread, circuses, jobs, and culture. Historical records, archaeological remains, and inscriptions on tombstones provide evidence of the migrants' experiences. There is no complete imperial-era census for the city of Rome, but historians have suggested about 5% of migrants were voluntary, while about 40% were slaves. We could learn more about their lives from human skeletons if we could specifically identify the migrants. If we knew which skeletons belonged to immigrants, we could ask questions like who immigrated to Rome? Where did they come from? How did moving to the city change their life, their behavior, and their customs? Chemistry to the rescue. Strontium exists naturally in rocks and gets into drinking water when the rocks weather. Water is made of oxygen. Both of these elements, strontium and oxygen, exist in different forms called isotopes. Each place on Earth has a specific ratio of the different strontium and oxygen isotopes. Human body tissues preserve the elemental signature of the places we live when the tissues are formed. By comparing the strontium and oxygen ratios in a skeleton with the local environmental signature, archaeologists can tell whether someone grew up in the area or was an immigrant. Dr. Christina Killgrove used the teeth from 105 ancient Roman skeletons, 32 kids and 73 adults, that were buried in 2 different cemeteries in the city of Rome, so she could analyze the strontium and oxygen isotopes and learn whether these individuals were born locally or were immigrants to the city. Dr. Killgrove studied isotope ratios in skeletons from 2 different burial areas along the roads leading into Rome. What did the isotope data indicate about immigration to ancient Rome? There was a lot of variation in the population, which may mean that many of these individuals migrated from surrounding areas to live in the city. 8 individuals had isotope signatures distinct enough to suggest that they were immigrants from farther away, perhaps the Apennine Mountains or North Africa. The immigrants were adult males and children from the necropolis burials, suggesting that they might have had lower social status, or they may have been slaves. In a study of carbon isotopes, the researchers also found that after migrating to Rome, people experienced significant changes in their diet. This study is an initial attempt to understand immigration in ancient Rome. Eventually, this work may flesh out a lot of additional detail about the Romans who were left out of historical records. More research is needed to understand the impact of the Roman aqueducts on the oxygen isotope signatures. Also, analysis of skeletons could tell us more about the lives and lifestyles of these immigrants. We might find skeletal evidence of different habitual behaviors or work activities between immigrant and local individuals. There might be differences in skeletal markers of malnutrition, dental health, and other aspects of health that are informative about the experience of slavery in Roman society. Slaves and immigrants contributed labor, genes, and new diseases to Rome. In return, Roman society and culture shaped the lives and the bodies of these immigrants. Telling their stories using osteobiography and bioarchaeology will help historians and classicists understand more about slavery in the empire and will enhance basic knowledge about this extremely important and influential period in European history. Want to read the academic paper? Find it at PLoS ONE. This research was funded by the National Science Foundation, Wenner Gren, and the University of North Carolina at Chapel Hill. Video production by bioarchaeologists Speak.